thoughtful visualizations have the power to impact science by helping researchers to untangle complex questions. Questions like, how do you compare a human and a lizard? My collaborator, Manfred Grebherr, is tackling this question by comparing the human genome with that of the lizard and looking for regions of similarity between the two. Ultimately, he hopes these sorts of comparisons will shed light on how our own genome encodes who we are. Now, when Manfred and I started working together, he was struggling to make sense of his computational results using off-the-shelf visualization tools like this one. Now, in this plot, we're looking at one human chromosome against one chromosome from that of the lizard. And each one of those dots inside of the plot indicates a location of sequence similarity in these two chromosomes. But now, for Manfred to actually look across his full data set, he had to look at many of these plots. In fact, his whole data set was producing over 300 of them. Now, not only were these plots unintuitive and overwhelming, but they also masked some critical subtleties in his computational results. Now, he and I worked together for several months to design a new interactive visualization tool called MISBE. Here, I'm showing you just one part of that tool. MISBE is designed to support data like Manfred's, data that is complex, that's multidimensional, and multi-scale. And it was the first tool to fully support this type of comparative genomics data. Now, in this view, in the outer ring, we're looking at the human genome. And on the inner ring, we're looking at that of the lizard. The inner ring also contains one user-selected chromosome from that outer ring that is then emanating a set of colored lines. And each one of those lines represents a region of similarity between the two genomes. Now, using MISBE, scientists can quickly explore their complete data sets and build an intuition about the information contained therein. This is actually a screenshot of the very first data set that Manfred loaded into MISBE. Now, by some measures, this is a relatively aesthetic image. There's lots of colors and there's circles. Everyone seems to love circles. But for Manfred, this image was both shocking and disappointing, because what he saw was very ugly, messy data. He actually had no idea that his theory was producing so much noise, that there were so many lines going to different places. And this went against what he knew to be biologically true, and had been all but hidden in those dot plots. So he went back and spent a couple of weeks tweaking things in his theory, and he was able to get but this far. At this point, he scrapped his theory, decided to take a new approach, and that new theory that came out of that gave him this data set. Now, he's since gone on to publish his results and to release his software out into the scientific community. I asked him how long it would have taken him to make this breakthrough using the methods he had available to him prior to MISBE. What he told me was, honestly, I don't think I would have ever gotten here. Now, visualization tools like MISBE are actually a relatively simple outcome of a much more complex and messy process. Designing visualizations for scientists is more than just about creating images. It's actually about acquiring a deep understanding of their problems, of their questions, and also of their mental models. And it turns out that all of the hard work happens long before you write a single line of code. But when done well, these visualizations have the power to not just support these scientists, but to also influence them. But to do so, we have to move beyond thinking that visualization is just about pretty pictures, and to instead embrace that it is really about a deep investigation into sense-making. Thank you.